Drugs can be used to both relieve symptoms and to help cure diseases. In this video, we're going to discuss the process of development and testing that drugs must go through before they can be used in the real world. Luckily for us, microorganisms and plants have been evolving for millions of years, and along the way, they've evolved to produce a range of substances that are able to do all sorts of things, including killing pathogens. This means that instead of having to develop all of our drugs from scratch, we've been able to take substances from them and either use them directly as medicines, or sometimes modify them in a lab first, and then use them as medicines. Let's take a look at 3 examples to show you what we mean. Aspirin is a common painkiller that can be used to lower fevers. It's now super common, but it was originally developed from a chemical found in the bark of willow trees. Meanwhile, digitalis, which is used to treat heart problems like heart failure, was originally developed from a chemical found in these pretty plants called foxgloves. In fact, this finding originally made the plants so valuable that they almost went extinct in the wild. The most famous example, though, is the origin of penicillin. Alexander Fleming was a microbiologist who studied bacteria. One time when he returned to his lab after a vacation, he noticed that a fungus was growing on one of his petri dishes, and oddly, it had killed the surrounding colonies of bacteria. After investigating, he found that the fungus produced a substance that was able to kill the bacteria, and because the genus of fungus was Penicillium, he named the substance penicillin. And that's how we got our first and most widely used antibiotic. Regardless of how you find it, though, once we have a substance that we think could potentially be a new drug, we need to start testing it. There are 3 main things to keep in mind when testing drugs: efficacy, toxicity, and dosage. Efficacy is how well the drug works, so how well it produces whatever effect you're looking for. For example, how good is an antibiotic at killing bacteria? Or how well does a pain relief medication reduce your pain? A drug's toxicity, meanwhile, is how harmful is it? For example, does it damage our cells or have any side effects? Dosage refers to how much of the drug or what concentration of the drugs should be given. Generally speaking, the more drug we give, the more effective it's going to be. But it will also cause more side effects. So now that we know what we're looking for, we can look at the 3 main stages of drug testing. In the first stage, the substance that we think is going to make a good drug is tested on human cells and tissues, which can be grown in a laboratory. The benefit of this is that we can easily and cheaply test tons of different substances. The downside, though, is that this doesn't really tell us much about how the substance would affect an entire organism, or even how it could affect a particular organ. If the substance looks promising, though, then we can move on to the next stage, which is testing on live animals. For example, in the US, the Food and Drug Administration, or FDA, requires animal testing to ensure the safety of many drugs and devices. If researchers find that a drug is safe and effective through animal testing, they can begin testing it on a small group of people. Clinical testing in animals such as rats, dogs, mice, rabbits, guinea pigs, monkeys, hamsters can all give us a rough idea about the efficacy and toxicity of the substances in humans. These first 2 stages are considered to be preclinical because they don't involve any humans. The final stage is clinical testing, where we finally give the drug to humans. The first step of clinical testing is to give the drugs to healthy volunteers starting with a very low dose, just to check it doesn't cause any problems. Then we slowly increase the dose, with the doctors keeping a close eye on the health of volunteers, checking for any side effects. The aim here is to basically find the maximum dosage that we can give before we start to see side effects. If this all goes okay, then we give the drug to people suffering from the particular illness that the drugs are trying to target, and again slowly increase the dose. This time, though, we're looking for the optimum dosage, which is the dose at which the benefits are maximized but the toxicity is minimized. The last thing we need to talk about is how we can ensure that the process is fair and the results we find are valid. To achieve this, we see that clinical trials should be blind and use a placebo. A placebo is a substance that is just like the real drug, but doesn't actually do anything. For example, it will often look exactly the same, except it's composed of sugar instead of any real medication. The most common technique is to give half of the volunteers the real drug and the other half of them the placebo. But importantly, we don't tell them which they're taking. This is why we say that the trial is blind. In fact, most trials are actually double-blind, where neither the doctors nor the volunteers know which drugs have been taken by whom, until all the results have been analyzed at the end of the study. The purpose of running blind and double-blind studies is to avoid introducing unconscious bias. For example, if the volunteers knew that they were taking the drug, they might be more likely to report any side effects, and their doctors might be more likely to notice them. Finally, once all the drug testing is complete and the results are written up, it will be published for peer review in a research journal. When research is peer-reviewed, this means that the experiment, the results, and the conclusions of the study have been analyzed by other scientists to check that the tests were fair. This rigorous analysis by other scientists is central to all of science, and that helps to minimize the chance that false claims or results are published.